Thank you, Dr. Lang. Again, let me express my thanks to Dr. Valdivia and Dr. Sinning for organizing this. It is an incredible effort, and I'm really happy that we are able to come together in this. Let me project the screen here. I hope you're able to see the screen well.
Right.
Yes. Perfect. Okay, excellent. So the talk here will be about update on current standards as well as clinical trials that are going on, but I'll add a little bit about the preclinical work that we are doing as well. So as in the United States, when we look at our registries, we can see that glioblastoma predominates as one of the most aggressive of our tumors. And hence, a lot of our clinical effort in MD Anderson and many of the brain tumor centers focuses on trying to get more information about glioblastomas. But glioblastoma has many changing faces. And you heard from Dr. Fuller much more in detail. At one point, it used to be vascular proliferation and pseudopalisading necrosis. Then it was okay to just have vascular proliferation. Then the molecular things began to arrive. So there's IDH wild type or mutant glioblastoma. And you also heard how a mutant might be a glioblastoma, even if it's an astrocytoma, if you had molecular loss. And then in the glioblastoma, there could be MGMT unmethylated or methylated classifications. And then there are new things that have emerged, such as epithelioid glioblastoma, histone mutant gliomas, which act like glioblastomas, and astrocytic tumors, which are IDH wild type with specific molecular changes. So what does this mean? You're seeing there's an evolution from histology to genetic and epigenetic insights modifying the diagnosis. And I'm very sure, and I'm sure Dr. Fuller would agree with me, that we are not at the end of the road yet. I suspect we may get many more subtypes of glioblastoma going forward, which might be extremely important in deciding what treatments to offer to the patients. So if you look at the old trials, this is a summary of many trials that were done, and you can see a number of patients, over 1,600 patients here that were looked at, compared with 1,300 who had radiation therapy alone. The question here was, does chemotherapy add anything? And this is a meta-analysis of multiple trials by Stewart and colleagues. And what they found was, if you look at the median survival, there was barely a difference. If you look at a 2-year survival, maybe some difference, but it was very modest impact of chemotherapy until this time in 2002. When BCNU wafers were introduced, there seemed to be a little bit of a push, but again, you can see the difference is so minimal that people were not very enthusiastic about this, although there was a tail effect. It should be noted that this was high-grade gliomas in general with a subset of these patients, the majority actually with glioblastoma. 2005 changed a lot of things because that's when Roger Stupp and his colleagues from URTC, NCIC Canada published this paper where they compared radiation therapy with temozolomide, with adjuvant temozolomide against radiation therapy alone. And strikingly, it should be remembered, these were for patients who were less than age 70. There was an improvement in the combination versus radiation therapy alone. And if you looked at the overall, so this is the progression-free survival. If you look at the overall survival, the median survival improvement was in the order of about 2.5 months. But the 2-year survival was quite striking, more than double the people were living longer, and it stretched out. And later analysis showed that that remained separate for a long period of time. What was even more striking is for the first time, we saw that MGMT, promoter methylated tumors, patients who had that had almost half of those patients were alive at 2 years. And that continued on for a long period of time. At 5 years, there were survivors, and there are even some 10-year survivors. That was unheard of in glioblastoma. So this truly marks a landmark publication and effort that changed the perspective on glioblastoma. I think one of the biggest things it did was to change the perspective of this being a terrible tumor that you can't do anything about. Now oncologists were entering into the picture to say, maybe we can treat this with chemotherapy. Very unexpectedly, there was additional studies done with this electrical therapy, where you put the electricity pads on the scalp with ceramic discs like this that are connected to the battery. And that in turn pushed the survival even more. So you can see the median survival pushing by about 4.9 months. And if you look at the 2-year survival, it's reaching close to half of these people. And these are unselected people, MGMT methylated and unmethylated. And going on to 5 years, you can see about 13% of the patients getting both of these treatments being alive versus 5%. And if you looked at the methylated or unmethylated tumors, it appeared to provide some benefit in either group. So suggesting that this was somewhat independent of the molecular markers. And this was a totally new type of treatment. And there are still controversies about people who either believe this works or think that this may not be really a good result. But the numbers are not trivial. This continues to evolve, and there are additional studies coming in, and we are trying to understand what the mechanism for this is. Now, if you switch, and if you notice, the previous studies were all keeping patients under 70 years of age. So there's another study that was done looking at elderly patients defined as above 65 years of age. And Jim Perry and his team conducted this study, and they showed that the combination of radiation and temozolomide, similar to the Stupp regimen, was actually also beneficial in these elderly patients compared to radiation therapy alone. And that result, about 33% decrease in hazard was highly significant, even though the numbers were not very impressive. You can see the median survival, even in this good prognostic group, is not very high. But again, what they noticed was whether it's unmethylated or methylated, there was benefit at the 24-month mark, suggesting that in this group, the treatment helped both types of patients. Now, I want to switch gears a little bit to talk about the molecular subtypes that we noticed before. So histone mutant gliomas are somewhat new. And what it refers to is this protein that has this tail, which is the basic protein around which DNA strands are bound. And there are many different types of histones. This particular one refers to histone H3, which has these tails. And specifically, if you look at K27, G34, and K36, these are the key players. These are methylated, and that methylation is really important in their function. So when the G34 is very close to K36, so if this is damaged, that does not work. Whereas here, any direct damage makes this not work. So in the K27 mutant tumors, what was found was when the mutation happened, the partner proteins that interacted with this actually stopped working, even in the surrounding areas. So that stopped many of the processes that would normally have been conducted. In the same way with the G34 mutation, this one showed the same kind of blockage, causing decrease in the methylation of these residues when this becomes damaged. More interestingly, this was seen mostly in young patients, and in the pediatric and young adult patients, they mainly happened in the thalamus, the pons, and the brainstem. Whereas the G34, the second mutation, seemed to be more hemispheric. And each of these locations had some associations with other mutations. What is the impact of this? That is the mutant glioma, even in young patients. Terrible prognosis, very poor survival, as you can see, whereas the wild type seemed to be better. And you can see that in wild type glioblastoma, if you take a H3K27 wild type, you can see the survival of that is better than the mutant, even if it's a midline glioma not classified as glioblastoma. And it doesn't matter where the location of the tumor is, you can see how all of those tend to be very poor. And from that perspective, many attempts have been done to treat this tumor and very little success. One of the newer ones that has come into attention is ONC or ONC201, which is a dopamine receptor D2 inhibitor, which is a strange target for a glioma, but it showed significant responses in some of these patients. And you can see in this publication that there are patients who are still doing well on this for a length of time. Considering the survival is very poor, these were considered to be very impressive, and this is actually being reviewed by our FDA for possible approval for this disease. The other molecular alteration I want to talk about is the IDH mutations, and the most common mutations are 132H, and it predominantly happens in 2 to 3 grade 2 or 3 gliomas, but there is a subset of secondary glioblastomas that often evolve from these that carry that as well, compared to the primary glioblastoma, which does not have that. What is interesting is, again, switching from histology to molecular markers. This is glioblastoma, IDH wild type or IDH mutant. And if you take the anaplastic astrocytoma with the same kind of category and superimpose this on this, you can see that the IDH mutated GBM almost behaves better than the IDH wild-type anaplastic astrocytoma, bringing to focus the point that the histology might not define anymore the behavior of the tumor, and that molecular markers may be important. And those survival things here are noted in these survival numbers. So here we are with a series of studies, and you can see if you look at the old days and then compare it to the more recent days, certainly we have had some improvement, but there is much more to be done. And the question is, where do we go from here? Are we forever trapped in this 3 to 5 year range, or can we lift the survival up further? So much more work to be done. I will briefly touch on recurrent glioblastoma, which has maximum feasible resection if the patients are symptomatic or if diagnosis is necessary in very selected patients. But otherwise, in the United States, we often use bevacizumab. In some treating physicians use Novo-TTF, the electrical treatment, but that's very limited now. In Europe, Novo-TTF is one of the things that's used. Bevacizumab is not approved yet. In Asia and other places, nitrosoureas of different kinds, temozolomide, bevacizumab has been approved in some countries. It's now gaining more popularity. In Latin America, best as I know, it's still CCNE or bevacizumab, and there are some other agents that are local that are used as well. There are other agents such as CCNU, 13-cis-retinoic acid, dose-intensive temozolomide in the MGMT methylated group, or Avastin plus other drugs. But most of these, none of these have shown improvement in survival, so new treatments are desperately needed. And this is one of the things that looked at Avastin, and you can see again the survival, although it was not improved, there was with or without the addition of chemotherapy, there was no difference. There was improvement in the overall response rate, and there was improvement in quality of life, and that is the reason this was approved. But there is a lot of criticism about that approval. I want to switch gears and talk about the reasons why we have all of these problems with this drug. The blood-brain barrier is certainly a big issue. The pharmacology of the drugs is a big issue. So pharmacokinetics, drug-drug interactions, efflux pumps that throw the drug out of the cell. So you give the drug, it goes into the cell, it gets thrown out. That can be a big factor. In fact, if you look at erlotinib, which is one of the drugs that you use against EGFR mutant tumors in lung cancer and other tumors, but in glioblastoma, when you use this, if you looked at the wild type thing, very little gets in. If you knock out these efflux pumps, which are throwing out the drug, then you can suddenly see that the drug gets in much better. And if you look at all the drugs that have been targeted therapies that have been used in this treatment, you can see a number of these are thrown out of the cell by these drug efflux pumps. So we need to pay attention to these kinds of things in order to say why the drug does not work. It may not always be the case that the drug is not working. It may not even be getting in there. That brings me to the fact that we look at tumors now very differently from histology. We are looking at the gene expression profiling. We have classifications of subclassifications of these glioblastomas. We are beginning to see there are different patterns. Roll-Verhoek did this landmark study in 2010 that suggested there were many subtypes of glioblastoma which could be identified by markers. In these analyses, they also identified multiple pathways, but you can easily see this is 77, that is 86, this is 8. So this is not exclusive. These are all overlapping pathways. So these are complex tumors with multiple interactions between these pathways. So in addition to the genetic signature, there are epigenetic signatures related to methylation. And here, for example, you can see in GBM studies by Noshmer and colleagues that there is a distinct population here that seems to be completely different from the others. And that turned out to be a subset of the proneural glioblastoma, what you saw in the previous slide, that had much better survival compared to the proneural non-GCIMP, the ones that don't have the methylation, that has almost the same survival as the rest of the tumors. So clearly identifying these kinds of subsets is very important in terms of both classification as well as looking at more details of these tumors. Now, Capra and colleagues did a much more extensive methylation analysis, and they were able to show clusters that came from the methylation analysis where tumors seem to come together. So you can see some of these tumors like glioblastomas mesenchymal, glioblastomas midline. There are receptor tyrosine kinase type of glioblastomas. These clusters are very different from, for example, astrocytomas, which are IDH1, or medulloblastomas, which are a completely different type of tumor. So you can see how this spreads out and reclassifies. And what actually happened in this particular study is you saw many of the tumors that were by histology classified by certain names had to be reclassified, many of them as glioblastomas, from the original diagnosis using this particular criteria. So there is clearly more to be learned, and our histology is very limited in terms of identifying these types of subsets that may have different prognosis. A very strong effort by three predominant bioinformaticians, Dr. Roel Verhaak, Dr. Antonio Ibarrone, and Dr. Hutan Nushmer, resulted in this fantastic study where they looked at both gene expression and methylation and then overlaid all that to find out what are the subtypes. And there are many things you see here, but I wanted you to focus on the fact that in IDH mutant tumors, which are generally thought to be better prognosis, there's a subset whose survival is as bad as the other glioblastomas, suggesting that if we identify these patients, they may behave differently from the rest of the patients. Similarly, if you look at the typical glioblastoma-like signature, which are IDH wild type, there is a subset that seems to have good prognosis compared to the rest. So these subsets are now beginning to be identified. It is not in clinical practice. There are practical difficulties in doing this, but that's still steadily moving towards that arena. The other interesting thing was if you took one tumor and then analyzed the cells in there, turned out that these subtypes that we talked about, whether it's neural or proneural or classical, they are not different types of tumors. Within the same tumor, you could see these classes coming in. So that suggests that tumors have many different types of evolutionary subtypes, and these subtypes can shift within the tumor, and that can result in these changes that we see. So it's a very dynamic situation, not a static kind of picture. In addition, this Korean group did a study where they looked at ALA, which is a fluorescent guided standard that can be used for surgical resection that I think you will hear about more later in the session. But what they did was they took the tumor and then took samples from different parts of the tumor and studied them to see what types of cells there are in these areas. And what they found was there were some cells that looked like the starting of the tumor, and then they seemed to diverge and evolve into these different types. And what was initially a proneural tumor, later in one spot became a mesenchymal tumor, suggesting that there is this evolution even in this space as the tumors grow. And this is an important thing because that suggests that these tumors are not just going to be one name or one type. There is many variations in that. That even affects treatments. For example, these are all cells taken from one tumor and then treated with different drugs. And what you see is even within one tumor, there are some cells that are sensitive and some cells that are resistant for multiple different types of drugs. And they all look somewhat different if you begin to grow them up. So very significant intra and intertumoral heterogeneity. Now, when you look at all these mutation things, and if you look at the immunologic parts of this tumor, you can see that there are several immunologic characteristics that are very interesting. And based on that, there have been many immunotherapy studies that have been done. Here I show you some immune checkpoint inhibitors, multiple combinations. Many of them have not succeeded. They have not really improved the outcome. The second set of things that I wanted to share with you is about vaccines, and there are many different types of vaccines. This, for example, CGFR-V3 vaccine led by MD Anderson by Dr. Heimberger. Unfortunately, in the phase 3 study, did not show benefit. We have CAR T cells, now CAR NK cells that are being tried using specific targets. This requires specific tumor targets, and that's been the challenge. And finally, oncolytic viruses or biologic therapies. And Dr. Lang, Dr. Fue, and Dr. Gomez-Manzano have led the effort in MD Anderson looking into this, both by giving it straight into the tumor and more recently through cellular therapy that can deliver this into the tumor. A couple of interesting points I wanted to bring up is Dr. Closey and team from UCLA found that if you gave the immune checkpoint before surgery, inhibitor before surgery, and then you continued the immune therapy, that those patients seem to live longer than the people who got surgical resection followed by the pembrolizumab or the immune checkpoint. And you can see here, if you look at the survival, these are the people who got the neoadjuvant pembrolizumab, and these are the people who got the adjuvant pembrolizumab. So that difference was quite striking in this small study, but it's a small study. So there are larger efforts that are going on, and they suggests that the immune suppression in these non-resected tumors versus immune activation in the resected tumors was perhaps the reason that that happened. I want to briefly talk about the clinical trials here that are ongoing. There are many drugs and many radiation combinations and immune plus drug combinations that are going on an ongoing basis, and all of these have literally, are continuing, and they are now delving into the insights that we are getting from molecular biology in order to make this work. So these are the challenges we face. The fact that there are glioma stem-like cells that are resistant, that there is tumor heterogeneity, that there's blood-brain barrier and immunosuppressive microenvironment. In the interest of time, I'm going to skip a few of these slides, which I was going to talk about, but I want to get to the last point, which is really the attacking this from a different direction. So energy utilization in gliomas is a very important part, and we are finding that there are certain compounds, certain proteins such as NAMPT, which help produce this energy in the cell. And people in my lab have been working on this, and we are finding that inhibiting NAMPT has caused significant decrease in many of the characteristics of glioblastoma, and it can affect the stem-like behavior that can be resistant to treatment. We have also shown that we can shut down energy utilization, and that has resulted in a drop in these cells that are glioma cells, but it does not affect the normal cells as much. So we hope we'll be able to target this. And we have many new treatments that we are working on, different targets that are completely agnostic of the genetic susceptibility. We have set up a new program, which is headed by Dr. Lang, Catalyst, which collects tissue and studies them, but we are also now launching a program for development of drugs. And this pipeline will then hopefully get us new drugs that we can take to trials. So in summary, I wanted to just mention that current standards have very modest efficacy and novel therapies have really not worked. And the tumor heterogeneity is a major cause of resistance, and we don't understand this fully. And we need better preclinical models. We have to tackle the complexity of this and then begin to translate these findings. So with that, let me stop and thank you all for listening into this talk.